DNA repair, I wanted to go back and make sure that this part was clear and the previous recording, especially I've missed some of the notes at the time. So what we see here is arrangements of genes as we go in terms of reference into evolution as a mechanism, something I've mentioned. Then, but what I'm going to make it clear is, as an example here, say we have an early globin gene, and then through the process, gene duplication takes place. And this gene duplication is going to impact, so we will have alpha gene, for example, and then the beta version of that gene. Now, that gene could be subject into certain changes, such as mutations, as I've mentioned as well. And what I make clear here is homologous. They can be perceived as similar genes or proteins because they share the similar ancestor. Okay, and when we look into orthologs, there are two genes in two different species. So this is human, frog, mouse, two different species, but they share the common ancestor when you look at that. Okay, and eventually paralogs are two genes in the same genome. Okay, there are two genes in the same genome. However, they are the product of that gene duplication process, so they can be traced to that gene. Now, what I want you to do as a quick exercise, I want you to search and share with me an example of ortholog and paralog genes and homologs. Okay, so I want you to give me a specific example of a gene name that's defined as or genes that are defined as orthologs, genes are defined as paralogs, and genes are defined as homologs. Okay, and you would share your again answer with me. Other thing, as I'm gonna move here now, also to make sure you capture that understanding, we need to look into chromatins, chromatides, and the different processes of cell division. Here's one question for the chromatides, okay? Just to make sure this is not confused with chromosomes, here's my question for you. When chromatides take a place, or they make sense to be referenced or used as a term, okay? In other words, are chromatides part of the formation of the chromosomes, or they are already there in our DNA? So when we look into this shape here, do we need to refer to it as just the karyotype representation, a depiction that we have of the chromosomes? Chromatides is not part of this, or is chromatide part of that, and at what time? And to simplify the question, all I want to know from you is at what time chromatides appear, okay? Chromatides appear, and your understanding of chromatide is going to make it clear, and then it's going to be one key answer. This is what I'm looking for, and in fact, I gave you the answer in my brief revision here. Just want to make sure some of the concepts are clear for all of us. So we went with the mutation, we discussed different types of mutations, diseases, and processes, and their categories. Now, one of the interesting concepts here is when mutations are part of the biological mechanisms in our cells, of course, you can imagine some of the mutations can lead to certain diseases, as we have looked into previously, like minor mutation can cause major disease. So there is a very important process in our biological system that is referred to as DNA repair. And in fact, there are a set of mechanisms as part of the DNA repair mechanisms. I want to break this part down a little bit because this slide is taking us directly to the following. DNA repair mechanisms, although they are intended to fix mutations, they themselves can induce mutations. So it can be a sort of confusion, okay? But let's break it down and summarize some of the ideas that want to reflect here. And if like, you know, one of the things that I brought into your attention earlier, it is one of the common reasons for why there can be some mutation. Deamination. It refers to removal of an amino acid group, okay? Removing an amino acid group from a molecule. So in general, that is in general definition, okay? Now, in terms of DNA, when we look into DNA as a concept, it is this conversion of one base, conversion of one base to another, okay? And I refer to this earlier when you say deamination happens during DNA replication. There are lots of interesting details there, and how eventually things like number of cytosines subtracted from number of guanines as a distribution window over the DNA can lead us to recognizing certain shifts that may hint the positions of origin of replication. But anyway, what I want to here to highlight is, if you remember, we were looking at the forking stage of the DNA replication. Mutations in general can take place on the leading strand, right? And can take also place on the lagging strand. There is a higher chance in the lagging start because these Okazaki fragments that we have discussed earlier take a little bit more of time until they are there's more involved process until the whole new sequence is created. So there is a slightly higher chance for some mutation here. But I use it as an example because it may give you this idea, okay, we are creating a new piece here of strand, but we know DNA likes to be double-stranded and bound to each other very strong. And now this is one-stranded, and this may cause issues of instability. Eventually, some mutations are going to happen. So I want to make it clear why it is part of the process that some deamination happens. For example, one of the very common examples is the conversion of cytosine into uracil, okay, in the DNA, which is not typical component of DNA. It is a typical component of RNA, uracil, I mean. So if it is not repaired, it is gonna, if it is on this strand here, and if it is not repaired, what's gonna happen if it is not repaired? As the process goes on, this is gonna be paired with adenine, okay? So remember, originally this was cytosine, and actually it should be guanine. This is what should be, not adenine. But since uracil happened because of this spontaneous kind of mutation, again, it's going to end up with the results of having uracil adenine base pairs instead of cytosine guanine. So in the next round of replication, this is going to be fixed, okay? And look into this. I mean, there is going to be a DNA replication process. So let's look into that. This is adenine. And how is that going to be fixed? Okay, well, it's going to be able to recognize, okay, this is uracil. So we shouldn't have a uracil in our DNA. So this is going to be changed into thymine. Okay, thymine. So we end up with TA base instead of a guanine and cytosine. So this is of course a very clear mutation. And in fact, even the DNA repair in the second round, when there's going to be a DNA repair mechanism, is not going to be able to recognize easily this the source of this. So this was going to do, and we are going to end up with a mutation. So this discussions introduces us to why we may have deaminations or some mutations, and how DNA repair is very important process, but sometimes it does not guarantee that we will have everything settled for us as we are looking into. And in terms of as well, DNA repair mechanisms, there are many DNA repair mechanisms, okay? DNA repair mechanisms. There are many DNA repair mechanisms. Apology, give me a moment. Okay, so one of these repair mechanisms is what's called direct reversal. So some DNA damages can be directly reversed, okay, direct reversal. We'll look into this in the slide. There's what's called base excision repair, base excision repair, nucleotide excision repair, and so on and so forth. Mismatch repair, and there are repair mechanisms related to double-strand break in the DNA, like non-homologous end joining and homologous recombination. Now, let me highlight one of the examples in a little bit more details. That is, for example, let's take base excision repair, okay? BER for short, okay? Base excision repair, okay? So if there's a spontaneous change, for example, that same conversion of thiamine into uracil, which is not normally found in DNA, it is an RNA base, so this is an error, okay? So that error is going to be recognized by certain enzyme. So if I give here an example, let's say we have these two strands and we have some bases, okay, that are paired to each other, okay? And let's say this is the region of error, okay? Okay, let's say that this is the region of the error. So there's what's called DNA glycosylase. Let me write this down. Glycosylase. Okay. DNA glycosylase. Sorry, this should be an S. Sorry for that. It is a specific enzyme, okay, that recognizes the misplaced uracil. So there is a process that can come here and recognize after the whole DNA replication takes place. How it's going to do this? I don't know if I'm gonna be good in drawing some hands, and these are the muscles, okay? Gives you an example of some person here with good muscles, maybe a little bit fat. So it's gonna come here and destroy or cut this part, because this enzyme recognizes this shouldn't be here and it should be removed, okay? So it's gonna take this part, let me use, okay. Okay, so I'm gonna draw the line again. So we are using the eraser just to show you that this is gonna be taken away, and then we'll end up with this part, okay? There is this region now through the process of this enzyme called DNA glycosylase coming here and leaving behind what is referred to as AP sites, okay? It's epimeridic site. So for short, this is AP site, okay? It is this region that is now empty. And so there is what's called then an AP endonuclease. It is another enzyme called AP endonuclease, okay? This other enzyme is going to recognize the site and makes and it's going to actually do the following, okay? Well, I guess I've keep looking into make sure get this right. Yes, I think what I've the DNA glycosylase is not going to cut this region, but it's going to recognize this amino acid, okay, or nucleic acid, and take it away, okay, and this mechanism. So it's going to remove the base, okay, it's going to remove the base from this area. Now, AP endonuclease is the one that's going to cut this area, okay, is the one that's going to come and cut this area. So these are the specific roles of these two enzymes. Eventually, this is going to be a removed base, and there is going to be the AB site, and AP endonuclease is going to come and try to fix it, okay? So it's going to recognize the AB site, makes cut in the DNA, and this is going to prepare after there's a cut here, okay, and cut here. This there is a gap that need to be filled, okay. So there is this region here that we have to fill it with some information, because now this whole base pair disappeared, gone. It cannot stay as this. So we can imagine. Now it's going to be an interesting question. What do you think? Which enzyme is going to come here and start filling this region? Now, although there are lots of concepts and terms, hopefully this is one of the concepts that got stuck in your brain out of the previous lectures, specifically the DNA replication. So whatever, write down what you think. It is some kind of enzyme related to the DNA replication, which is going to be DNA polymerase. Okay, so DNA is going to fill this, and then at the end, ligase is going to come and do and going to seal everything. Okay, there are a bit of details like how it's going to be the ending here, and this is going to be also removed a little bit for this region. Anyway, so this is one example of one of the processes of fixing or DNA repair mechanism. And if we look here in the slides, as you see, there is different types of repair mechanisms. Okay, specifically direct reversal, for example. So there is an enzyme called photolyase that can reverse UV light-induced dimers when exposed to light. Okay, so this is one of the direct reversal mechanisms. It can reverse some of the affected bases. Okay, there is mismatch repair, okay, which I gave already a similar example to, as well as other types of mismatches that you can look and see in the slides. We have discussed also the endonuclease and the end polymerase as part of general roles in this. There are other types of DNA repair mechanisms like double-strand breaks and, for example, non-homologous end joining. And in this case, in the non-homologous end joining, in general, double-strand break repairs are dangerous. Okay, this is not one strand; it is now affecting two strands. So this is dangerous. Okay, for the non-homologous end joining, the broken ends are directly ligated together. Okay, so you can see this; those are going to come together through the DNA ligase. So what you see here, DNA ligase can be perceived as a sort of important role, but again, when it comes in play here, its own role, when it sees this kind of non-homologous end joining case, it's going to seal for us and bring together directly two broken ends. And this is can lead to insertions or deletions in the process. Okay, and there are otherwise different types like this homologous recombination, and it's going to use sister chromatide. Remember, I've asked you to report for me what is chromatide. Okay, or at what stage do we recognize that chromatide. And so it's going to use the sister chromatide to repair the break, okay, and so on and so forth. So in this slide, taking from one nice good read paper that hopefully you find it there accessible through the notes on the slide, what we see here, okay, is an illustration of the different DNA repair mechanisms, and specifically, let me highlight this. Mutagenic sources, okay? So there can be different sources of the mutations that can take place, okay? So you can, this could happen through some intercellular processes during replication errors. If there's a source, exogenous resource, so it is not inside us like indigenous resources, it is coming from outside like UV light and other kinds of types that may cause different mutations. So mutagenic sources, there can be different types. Either they are indigenous, meaning they are happening as part of our internal system, or they are taking a place externally, okay? Externally, meaning meaning in this case from other source, radiation exposure, and so on and so forth. Now, for each one, there is going to be some issue with the DNA for each of these exposures, okay, mismatch indels, and then there's going to be a repair mechanism, and for some repair mechanisms, you will see the paper discussing that there has been reported syndromes, okay, there has been some reported syndromes or diseases, okay, or diseases. So DNA damage actually arises consistently from these different resources that we have looked into as indigenous or exogenous, okay? And now, the DNA repair is certainly required for the genomic stability that we're looking into. However, deficiency in this repair, any deficiency in any of these repairs, as I gave you in that illustration, sometimes DNA replication is going to end up giving us, instead of fixing uracil, saying this is thiamine, and so on and so forth. Now, if this may happen, there can be some accumulation leading to some DNA damage and eventually some genomic instability, okay? Some genomic instability. This may increase the risk of cancer development, or there are several syndromes associated with defects in the DNA cross-link repairs. So you can look into these different syndromes or diseases that are the results, actually have been recognized and referenced to be a result of the DNA cross-link repair manifest. So there is a repair taking part of that process repair, but it is affecting the output eventually, okay? Now, this is another interesting figure here. I'm going to quickly highlight a few things here. Okay, this is a recent slide, very interesting read, actually, also as from the source. It is telling us that there are different types of mutations. Can we recognize these mutations? This is the question that we are looking at here. Yes, there are specific technologies like those employed by Pacific Biosciences. This is PacBio, or the recent, these happen to be developed in the latest few years, Oxford Nanopore technologies, as well as optical imaging. These are technologies that can help us in recognizing, for example, single nucleotide variants, okay? And for example, Pacific Biosciences developed this technology. It's called SMRT, or smart sequencing. It is single molecular real-time sequencing, okay? Single molecule real-time sequencing, and that is helpful for sequencing and observing the DNA synthesis in real time. So it is characterized by long reads. It helps in recognizing intra-read variants and inter-read variants. So across the different DNA regions. And so intra-read is going to focus on long-read individual bases and small insertions and deletions, and there are large insertions or duplications. This is going to recognize that sort of part, like, for example, tandem duplication in this case. So there are these mutations that can take place at the level of the germline mutations or the somatic mutations. In case for the germline germline mutation, we know now it is either the sperm or the egg, and so the person can be a carrier, or sometimes this mutation can be seen and recognized through the effect on the somatic mutations on our cells. So specifically what we can refer to as chromosomal mosaicism, okay? And it refers to the presence of two or more genetically distinct cells population in one individual, as you see here, okay? So there are two kinds of population in this case. One of them is having some issues, like missing a chromosome, for example. A clear example for this is what's called vitiligo, which is one of the syndromes. It's a complex dermatological condition. It's not painful for the person, although it affects sometimes the emotions, and you would appreciate that. It affects the immune system and affects the colors that's gonna, for the pigments that are gonna eventually leave patches on the skin. So gonna stop here and then continue the next part.